Where is the mic? Good morning everyone. I am going to discuss about photosynthesis. So this is regarding to NEET, not for the IP exam. Okay, right. So photosynthesis is first. What are the questions will come from photosynthesis? It's a very important topic for NEET exam. Okay, in physiology, photosynthesis, respiration are the main things which are mainly important for NEET exam. Okay, so we have to more focused on these topics anamata. So coming to the photosynthesis, so earlierly photosynthesis means what? What is meant by photosynthesis? Definition of photosynthesis, you know, preparation of food materials by plants is called as photosynthesis, right? So here, what are the prerequisites for photosynthesis? Carbon dioxide and water are the requisites for the photosynthesis in presence of sunlight, and produces what? Glucose molecule. Glucose molecule, right? So instead of that, it releases oxygen and water molecule also outside. Okay, so these are the reactants, and this is the product. Okay, right. So CO2 and water molecule. How many CO2? 6 CO2 and 12 H2O. This is called as equation of photosynthesis. So equation of photosynthesis, what are the reactants? This may be a question. We don't know. Okay, and what are the products? What are the products? So before the photosynthesis actual process, there are some experiments are done by different scientists. Like you people know that you already studied it in your previous classes. Okay, Belger experiment and Half-Leaf KOH experiment, like that, right? So there are some scientists names will come, like Priestley. Priestley said that plants can repair the bad gas released by the animals. That means the bad gas which is released by the animals, which is cured. That means it is purified by plants by releasing oxygen anamata. By releasing oxygen. That is said by whom? Priestley. Okay. Later some other scientist called Ingenhousz said that oxygen is evolved in photosynthesis. By observing some algal blooms, where the algal blooms are there during the sunlight time, there are some bubbles are coming from the water. That indicates that it is the O2 is result of photosynthesis, said by Ingenhousz. And next, Van Sachs said that some glucose is accumulated during photosynthesis. That means glucose is the product of photosynthesis first identified by Sachs. Okay, right. Next, Engelmann is the scientist who splits the light spectrum into Vibzior anamata. That means light spectrum is divided, and he conducted experiment on a algae called Cladophora, which is a green algae. He observed that during the photosynthesis of the Cladophora, some aerobic bacteria are accumulated mainly in blue light and red light. That indicates that aerobic bacteria, that means it need what? Aerobic bacteria need what? Oxygen. So that means oxygen is evolved in photosynthesis, which is more at which light? Blue light and red light. Okay, that is explained by Engelmann. Okay, and next, the scientist Van Niels given the equation of photosynthesis. And definition of photosynthesis. That means in a suitable oxidizable compound, it is reduced the carbon dioxide into glucose. That is said by Van Niel. That is the equation called 6 CO2 plus 12 H2O gives rise to C6H12O6 plus 6 O2 plus 6 water molecule. Okay, and here if it is not water molecule, that for some of sulfur bacteria will be there. H2, yes. So here not the water molecule, here sulfur will be released out. Sulfur will be released out in the sulfur bacteria. Okay, sulfur bacteria, right? So this is the prehistory of photosynthesis. Prehistory of photosynthesis. Some of the scientists who are came: Priestley, Ingenhousz, Sachs and Engelmann. Okay, and next Neil. Okay, right. So here one more doubt will become, that is water molecule is there, here oxygen is there, CO2 is there, oxygen is there. That means here O2 is present in carbon and O2 is present in hydrogen also, right? So which one is donating this oxygen evolution? That means during the product formation, here the reactants both are having oxygen. Both are having what? Oxygen. So from which reactant the water is released out? That is the experiment done by some scientists, and by using O18 isotope. They used O18 isotope and given to this, first they given to O18 to the carbon dioxide. So in the carbon dioxide, carbon plus oxygen is there, right? So the oxygen is, which is nothing but O18 isotope. They are conducted the experiment, but here there is no O18 in this oxygen. That means it not from carbon dioxide, and later they conducted experiment with the water. That means in the water also oxygen is there. That oxygen is nothing but what? O18. O18 isotope is used for this reaction, and here in the formation of O2 there is a O18 is there. That means it indicates that what? Oxygen evolved in this process is coming from water, not from the carbon dioxide. Okay, oxygen evolved during this photosynthesis is come from what? Which compound? Water molecule, not from the carbon dioxide. So that is the thing, matter. Okay, so right. And one more question will come. Why 12 water molecules? Why not 6 water molecules? Okay, why 12 molecules means I will give you the clarification later. Okay, later, that means after some time I will give you. So these are the prehistory of photosynthesis. Prehistory of photosynthesis. Now we will go to the, what is the site of photosynthesis? Site of photosynthesis. What is the site of photosynthesis? Photosynthesis site is nothing but what? Chloroplast. Chloroplast is identified by Sachs. Sachs given the name of chloroplast. Right. So if you see the chloroplast structure, chloroplast is having discoid like structure, 2 membrane bound organelle, and inside that it is having piles of coins like structures. Those are called as what? Grana thylakoids. And these grana thylakoids are connected by stroma thylakoids, and inside it is having a matrix that is called as stroma. Okay, stroma, grana thylakoid, stroma thylakoid and stroma. And here some more information, like it is also having 70S type of ribosomes and single stranded naked DNA. Okay, right. So it is a semi-autonomous organelle. Right. So coming to this, grana thylakoid, stroma thylakoid and stroma are the main components for photosynthesis. Okay, right. So coming to this, this is the site of photosynthesis, where this photosynthesis takes place, here in the chloroplast. Okay. So then next, which are involved in photosynthesis? That means entire structure is involved? No. The grana is having pigments. Chlorophyll pigments will be there. That means light harvesting pigments will be there. They are trapping the light energy. What are they? There are 4 types of chlorophyll pigments are there. Chlorophyll A, chlorophyll B, xanthophylls and carotenoids. Xanthophylls and carotenoids. If you see the structure of chlorophyll A molecule, it is C55H72O5 and 4 Mg. Okay, coming to the chlorophyll B, it is C55H70O6 and 4 Mg. And xanthophylls are C40H56 and carotenoids are C40H56 and O2. That means here chlorophyll A is blue green color, chlorophyll B is olive green color, xanthophylls are yellow color, carotenoids are orange color pigments. Okay, right. And these are called as accessory pigments or shield pigments. That means they protects the chlorophyll A molecule anamata. Chlorophyll A molecule. And if you see the structure of chlorophyll molecule, it is having a porphyrin ring will be there. Sorry, right. So center of it is having, this is the porphyrin ring. Okay, this is porphyrin ring with a phytol chain. Phytol chain will be there, and it is just like that of hemoglobin, and instead of that hemoglobin, that is actually in hemoglobin iron molecule is there, but in chlorophyll it is having magnesium. So magnesium is the center ion of chlorophyll molecule. Okay, right? So these are about pigments: chlorophyll A, chlorophyll B, xanthophylls and carotenoids. Oxygenated hydrocarbons, xanthophyls are oxygenated hydrocarbons, not carotenoids. Xanthophyls are oxygenated hydrocarbons, whereas carotenoids are only hydrocarbons. So this is chlorophyll A molecule will be in blue green and it will be in olive green. Xanthophylls will be yellow green and carotenoids will be in orange color. Okay, so these are the pigments which are trapping the light energy. This is the site of photosynthesis and these are the pigments involved in photosynthesis. Okay, right. So coming to that, there are phases of photosynthesis. Right. So there are two phases of photosynthesis, light phase and dark phase. Light phase and dark phase. Why it is called light and why it is called as dark? Why it is called light means here it occurs in the presence of light. That means it needs light energy. So what is the thing happen in the light energy means here the energy is produced in this phase, ATP and NADPH2. ATP and NADPH2 are generated in light phase. In dark phase, that will be biosynthesized into glucose anamata. That means here the energy is utilized in dark phase. Okay, so ATP and NADPH2 are the energy forms. So it is called as assimilatory power. So assimilatory power is generated in light phase. That's why it is called as light phase. Okay, right. So why it is called dark phase? Dark phase means it is not mean that it occurs in night time. It means that it does not require light. Okay, so that's why it is called as dark phase. So here coming to light phase, ATP and NADPH2 are generated. How these are generated? Light is essential for this reaction. So where the light is trapped? Light is trapped by chlorophyll A molecule, right? Chlorophyll A molecule is trapping the light. So here see, this is the light harvesting complex. That means pigments like all the pigments, chlorophyll A, chlorophyll B, xanthophylls, carotenoids are there. So here this is called reaction center. If you take it as a reaction center, that is nothing but chlorophyll A molecule. This is very much needed, right? So this reaction center can absorb the light at different wavelengths. That means chlorophyll A molecule usually have two types anamata. they can absorb the light above the P700 and below the P700. Okay, what is 700? 700 is nothing but nanometers, that indicates the light intensity. Okay, so 700 nanometers, at the 700 nanometers the chlorophyll A molecule can trap the light energy. So depending upon that, it is divided into two types, PS1 and PS2. PS1 and PS2. PS1 is the having P700 capacity, that means it can trap the light energy at P700 or above the P700. Okay, PS2 is the reaction center which can trap the light energy above P680. So it is called as P680. P680. So what is the reaction center of PS1? P700 is the reaction center, and in PS2 the reaction center is P680. Okay, right. So you understand that two systems are involved in light reaction. Okay, PS1 and PS2. So what they will do? They will transfer the electrons from one compound to another compound. Ultimately it generate what? NADPH2 and ATP anamata. Okay, how it will be there? That will be explained in two ways. That is called non-cyclic electron transport and cyclic electron transport. Non-cyclic electron transport and cyclic electron transport. Coming to non-cyclic and cyclic anta. Where it occurs? We have to know. Non-cyclic electron transport is occur in grana thylakoid. Grana thylakoid, whereas cyclic electron transport is seen in stroma thylakoid. Stroma thylakoids. Ma, it is the question. Where the non-cyclic electron transport takes place? In grana thylakoid. Where the stroma thylakoids, that means where the cyclic electron transport takes place? In stroma thylakoids. So non-cyclic electron transport involved by both the systems PS1 and PS2 are involved in non-cyclic electron transport, whereas in cyclic electron transport only PS1 is involved, not PS2. That means PS1 and PS2 are present in grana thylakoid. So in grana thylakoid two systems are there, PS1 and PS2, whereas in stroma thylakoid only PS1 is present. Only PS1 is present. Okay. So these are the two systems involved in electron transport, non-cyclic electron transport and cyclic electron transport. Why it is called as non-cyclic means it is not cycle back to the where it is started. Okay, but here in cyclic means it is come back to the start point where it is started actually. So here PS1, what is the PS1? P700, and PS2 means P680. Okay, now we will discuss about non-cyclic electron transport. Why I am so fastly explaining means you know already. You already studied it and you already written the exam also. That's why I am going fast, because you need to get more and more points from that. So here NEET exam is like we have to capture all the things, and so we have to get the actual points and matter. Okay. Right. So coming to the non-cyclic electron transport, this is P680 is the reaction center where it accepts light excited electron given by pheophytin, and from pheophytin it entered into Q cycle, plastoquinone cycle, and next cytochrome B6F complex, and from that plastocyanin, and it is given to P700. It get excited, but some intermediate compounds will be there, and finally it is reaches to ferredoxin, and from the ferredoxin NADP plus is converting into NADPH2. Electrons are passing like this. So here light is captured, here also light is captured, right? So this is non-cyclic electron transport. In non-cyclic electron transport, first P680 absorbs light, it get excited, it get excited, and the excited electron is given to the first electron acceptor, that is called pheophytin. This is the bit. This is the bit. What is the electron acceptor in PS2? It is nothing but pheophytin is the first electron acceptor. Okay, right. Pheophytin. How? So now this 680 become oxidized or not? It released electron. So it comes to oxidation state. It need electron or not now? So now these electrons are given by water molecule. Water molecule. This is entirely takes place in thylakoid membrane only. You have to remember it. Okay. So this is the inner membrane where the oxygen is splitting. Okay. So now this oxygen has to be splitted, right? Oxygen split. Water has to be splitted. Water has to be splitted. So for water splitting, it also need H nu. It's nothing but what? Light. So light is splitting the water molecule. Light is splitting the water molecule. It is called photolysis of water. Photolysis of water. Water is splitted in the presence of light with the special complex here, that is called OEC complex. OEC complex. What is meant by OEC? Oxygen evolutionary complex. Oxygen evolutionary complex. In this complex, calcium ions will be there, and manganese ions will be there, not magnesium, manganese ions will be there, and chlorine ions also there. So this is the association of these ions which is forming OEC complex. In the presence of light, it is splitting the water molecule. Splitting the water molecule. Usually, two water molecules are splitted at one time. So it releases 4 electrons, 4 protons and 1 O2 molecule. So 4 protons, 4 electrons and O2 molecule is also released out. Okay, right. So these electrons are accepted by now P680. Okay, this is like every time it will take place up to light is there. Okay, so usually we are breaking the system. That means actually it is not the point to point anamata. We are seeing it at one stage of the photosynthesis. So we don't know that exactly first P680s get oxidized, how these electrons are transported. We should not to know. We have to know that water is splitted. How many electrons are released? 4 electrons are released at one time, because 2 water molecules are splitted at one time with the help of OEC complex, that will be accepted by P680. Okay, and again P680 get excited and releases the electron and come to the oxidation state. It is again accepting the electron. Like that, this will be continuous anamata. Continuous, right? So here after that, so here protons are released, remember it. Okay, so electrons are moved to P700 with the help of plastocyanin, and P700 is also accepting the light. At that time this plastocyanin is giving the electron. The P700 is in oxidation state and it accepting the electron and it get excited. Some intermediate compounds are there, but finally it reaches to ferridoxin. So what is the compound which is accepting the electron in P700? It is nothing but what? Ferridoxin. You have to remember it. PS2, what is the first electron acceptor? Pheophytin. In PS1, what is the electron acceptor? Ferridoxin. So from the ferridoxin, here one enzyme is there, ferridoxin NADP reductase. Ferridoxin NADP reductase anta, which is help in here hydrogen ions are also required. Okay, right. Ferridoxin NADP reductase, in the presence of this enzyme, especially there in grana thylakoid. This is grana. You have to remember it. Grana thylakoid. Okay, right. So in the presence of FNR, NADP is converting into NADPH2. NADPH2. For this production, it requires 2 electrons. It requires how many electrons? 2 electrons. Remember this point, okay? So, these like ultimately it reaches 2 electrons or reaches to the NADPH2. What is the assimilatory power? NADPH2 and ATP, right? So, now this NADPH2 is generated, right? Coming to the ATP. ATP is also generated here. How it means? After cyclic electron transport, I will explain. So, remember this point, like protons are released, right? So, here protons are released in the lumen of the thylakoid. This is the thylakoid lumen. So, see this is the figure. Idanmata. Okay. So, this is the lumen of thylakoid where the protons are released. So, here also from stroma side, protons are coming. And here also at Q cycle and at cytochrome B6F complex, some protons are released into the lumen side. Okay, right. So, remember this points. So, here questions will come like non-cyclic electron transport involve of 2 systems, PS1 and PS2. That is the one. And next, what is the reaction center of PS2? P680. Reaction center of PS1, P700. What is the electron acceptor of PS2? Pheophytin. What is the electron acceptor in PS1? Ferredoxin. Okay. And what is the end product of electron non-cyclic electron transport? Through electron passage. That means electron transport. What is that? NADPH2. NADPH2. With the help of what is the enzyme responsible for that conversion of NADP into NADPH2? Ferredoxin NADP reductase. Ferredoxin NADP reductase, which is there only in grana thylakoid. Okay, right? So, this is the process of non-cyclic electron transport. Now, we will go to the, so what is the OEC complex is just beneath the P680 anamata, not the P700. P700 is so far from the OEC complex. So, OEC complex is associated with which system? PS2. What are the compounds in OEC complex? OEC complex lo em untayi? Calcium, manganese and chlorine ions are also there. Okay, how many protons are released for 2 molecules of water splitting? 4 protons. Remember this. 4 protons. This is a question. How many oxygen molecules are released by, I am asking one tricky question like one water molecule splitting. How many oxygens can release? 1 nascent oxygen will be released out. It's nothing but O, not O2. Okay, nascent oxygen will be released. So, for conclusion also we are taking like 2 water molecules anamata. So, for splitting of 2 water molecules, what are the end products? 2 water molecules can form 4 electrons plus 4 protons plus O2 molecule. Okay, right? Got it? Now, we will go to the cyclic electron transport. రికార్డ్ అవుతుందో లేదో చూస్తున్నాను. I'll go for live later. Live వెళ్ళిపోదాం. Right. So, coming to the cyclic electron transport. Cyclic electron transport involved by only P700. It get excited and some intermediate compounds will be there. Ferredoxin, Q cycle, and from cytochrome B6F complex, and later plastocyanin. From plastocyanin it move back to P700. So, this is the ఇది ఇలా మనం వేసుకుంటే ఇలా వేసుకుంటే మనకు రౌండ్లో వస్తుంది. ఓకే, రైట్. సో, హియర్ there is no OEC complex. Light get exciting the P700. So, at that excitation it releases the electron, and released electron is accepted by ferredoxin and from the ferredoxin it moved to the Q cycle, and next to cytochrome B6F complex, next to plastocyanine, and from plastocyanins to come back to the P700. So during this process only ATP are generated, no NADPH2. Why? Because there is no FNR. FNR is not there. What is FNR? Ferredoxin NADP reductase enzyme is not there in cyclic electron transport. Where it occurs? Stroma thylakoids. Stroma thylakoids. Right? So in stroma thylakoids there is no such enzyme which can reduce the NADP into NADPH2. Okay, so that only ATP is generated, not the NADPH2, and there is no OEC and there is no PS2. So no OEC, no PS2, no FNR. Only one is there, P700, which is seen only in stroma thylakoid. So in grana thylakoid, PS1, PS2, FNR, OEC is there. In stroma thylakoid only PS1 is there. Okay, right? So this is cyclic electron transport. Cyclic electron transport. So cyclic electron transport is seen in stroma thylakoid. Cyclic electron transport associated with the formation of only ATP, not NADPH2, and again here there is no splitting of water molecule, there is no enzyme also which can reduces NADP plus. Okay, so this is about cyclic electron transport. So during this process at Q cycle and at cytochrome B6 site, some protons are released. Those protons are mainly what is the assimilatory power? NADPH2 and ATP, right? So these two, no NADPH2 in cyclic. So what about ATP? ATP ఎలా ఫామ్ అవుతుందో కూడా తెలుసుకోవాలి కదా. So we'll go this chemiosmotic hypothesis. Now we are not concerning with the electron transport. We are concerning only the ATP synthesis. That is only concerning about H plus ions. H plus ions. This is the thylakoid membrane where the different complex will be there. So this is PS2 which is associated with OEC complex, and from the PS2 Q cycle, plastoquinone cycle, next cytochrome complex, cytochrome B6F complex, plastocyanin, next PS1 to NADP plus NADPH2, right? So during the OEC complex splitting of water molecules, some hydrogens are released, and from the Q cycle also some hydrogens are released, and from the cytochrome B6F complex also some hydrogens are released. And during the formation of NADP plus into NADPH2, it requires some hydrogens also that are also accepted from stroma side. So how this Q cycle or from cytochrome B6F complex, the hydrogen ions from stroma are moved inside, right? They are dragged out from the stroma side only. So these all increasing the hydrogen concentration in the lumen, isn't it? This is the space in the thylakoid, that is called lumen. So in the lumen of thylakoid, hydrogen concentration is increased, and in the stroma side, as it is giving protons at Q cycle or at cytochrome B6F complex or at NADP plus, more hydrogens are dragged out from stroma to thylakoid. So here will be a deficiency will come, isn't it? Deficiency of H plus ions, and here more concentration of hydrogen ions are there. Here one this is nothing but osmotic complex, osmotic imbalance will be there or not? If you know the osmosis definition, movement of ions from higher concentration to lower concentration through a membrane, that is called what? Osmosis, right? So here the chemiosmotic hypothesis is nothing but explaining that here in the lumen the concentration of ions are more, it giving some force, that is called as proton motive force. Proton motive force. Proton concentration is increased in the lumen side, which has to be moved from lumen to stroma to balance the H plus ions, right? So this proton motive force ఎక్కువైపోయింది, H plus ion concentration ఎక్కువైపోయింది, ఇవి బయటకు వచ్చేయాలి, right? So here one channel is there, that is nothing but F0 F1 complex. This is also called as ATP synthetic complex. ATP synthase complex. F0 F1 complex is there. This proton motive force help in the movement of H plus ions from thylakoid lumen to stroma side, right? So they are coming out from the ATP channel anamata. ATP channel during this high proton motive force, a force వల్ల ఏమవుతుందంటే it converting the ADP into ATP. So here the what is the force which is forming ATP means it's nothing but proton motive force. What is the proton motive force means? Increasing concentration of H plus ions giving some force, that is called as proton motive force, which is established in the thylakoid lumen, which is helping the conversion of ADP into ATP. ADP into ATP through a specialized channel is there, that is called as F0 F1 complex. F0 F1 complex. So here the ATP is generated like this anamata. Understood? So because of the H plus ions concentration increased in the lumen side compared to the stroma side, there will be a osmotic imbalance will be there. To balance this, the hydrogen ions has to be moved out. At the time of movement, they pass it through a specialized channel, that is called as F0 F1 complex or ATP synthase complex. During the time of passage of H plus ions from these complex, there is a conversion of ADP into ATP takes place. So what is the force? Proton motive force, PMF, is the main thing which is generating the ATP anamata. ATP, adenosine triphosphate. Adenosine triphosphate is generated. Ultimately H plus ions are moved inside the stroma side to balance. Okay, so this entire process takes place in the presence of light, right? Here formation of ATP is called as phosphorylation. Phosphorylation. So here ATP formation is called what? Phosphorylation. It occurs in the presence of light. So it is called as photophosphorylation. Photophosphorylation. Understood? So photophosphorylation is seen in photosynthesis, whereas in mitochondria ATP is synthesized there also, that is called as oxidative phosphorylation. Oxidative phosphorylation. Here it is photophosphorylation because it occurs in the presence of light, that is in the during light phase, so it is called as photophosphorylation. Understood? So how this ATP is generated? So ultimately ATP వచ్చేసింది, NADPH2 కూడా వచ్చేసిందా లేదా? So what is the ratio? We have to calculate. So because it's a NEET, we need to know. It's a NEET, it need to know. need anamata. Okay, right. So 2 water molecules produces 4 electrons, 4 protons and O2 molecule and chepam, right? So here for every generation of one NADPH2 molecule, it requires 2 electrons. 2 electrons. The 2 electrons are coming from one water molecule. One water molecule. That means here I said equation 6CO2 plus 12 H2O molecule C6 H12O6 plus 6O2 plus 6 H2O. Here why 12 water molecules means here 12 NADPH2 are generated. Actually for passage of Kelvin cycle, that is dark phase, it requires 12 NADPH2 and 18 ATP. 18 ATP and 12 NADPH2 are required. So for generation of 12 NADPH2, it requires 12 water molecules because it releases one water molecule can release 2 electrons. One water molecule releases 2 electrons. So 12 water molecules releases 1 NADPH2. So 12 water molecules releases 24 electrons which are synthesizing 12 NADPH2. 12 NADPH2. That's why it is 12 water molecules. Understood? Why 12 water molecules means for every generation of 1 NADPH2 it requires 2 electrons. The 2 electrons are coming from 1 water molecule. So for 12 NADPH2 it requires 12 water molecules. Okay, right. So NADPH aithe cheppam. What about 18 ATP annam? 18 ATP. So actually it requires how many ATP? 18 ATP anta. 18 ATP means it's nothing but 2 water molecule produces approximately 3 ATP. 3 ATP generated. So here 12 water molecules produces 18 ATP. 2:3 ratio, 18 ATP. So this is the requirement and this is the generation. Understood? So that's why 12 water molecule. 12 water molecule. Understand? Hope you understand this lesson and we'll continue later. That means Kelvin cycle will be explained tomorrow. Thank you.